Hello and welcome to today's Texas Science Festival session addressing Alzheimer's with Laura Colgan and John Pierce. We're so glad you've joined us. My name is David Padifar and I am the Department Chair of Neurology and the Director of the Mulva Clinic for the Neurosciences. Um, I'm a physician and a scientist, and every day I'm reminded about how scientific discovery embodies hope for people and patients with neurological conditions. And so we're delighted to have two eminent speakers today. Um, I wanted to say first to please note that all participants will be muted and without video for the duration of the webinar. Because this is one of our Science Sparks events, each speaker will speak for about 10 minutes, and then we'll move to Q&A. Please use the Q&A feature by hovering over the bottom center of your screen at any time to submit questions you would like our speakers to answer. We also have any pre-submitted questions you provide, and we'll do our best to get through as many of these as we can. I would like now to introduce Laura Colgan, Associate Professor of Neuroscience and the recipient of the prestigious NSF Career Award, a really great prize for promising young faculty members. Laura studies how groups of neurons interact and coordinate their activities during learning, particularly in areas of the brain that are critical for memory formation, and gaining insights into diseases associated with abnormal rhythms, such as Alzheimer's disease. Laura, please.
I'm so sorry, but it's showing John's screen. Is there, and it doesn't seem to be letting me share mine. Okay, thank you. Okay. Okay, thank you for your patience. I apologize for that little blip. Okay, today I am going to be talking to you about an exciting new potential therapeutic strategy for Alzheimer's disease treatment. And just to briefly mention that Alzheimer's disease is a devastating brain disorder that causes the progressive loss of memory in patients. And there's several companies, including Elon Musk's startup company Neuralink, that are proposing to develop neural prosthetics for treatment of Alzheimer's disease. So what is a neural prosthetic? Well, the idea is that electrodes would be implanted into the patient's brains together with a computer chip, which would then allow the brain signals that are presumably representing memories to be recorded and deciphered by a computer to see if there's some way to recover memories that may have been fragmented in some way. So how do these brain signals that would be recorded using electrodes, how do they code information? Well, the human brain has about 100 billion brain cells, which are known as neurons. And each of these neurons sends a particular message related to your thoughts, your actions, your perceptions of the world around you, and your memories. And the way that neurons transmit information is that they actually fire off these little spikes of electricity that are called action potentials, or we do actually refer to these as spikes. And so this is a schematic to kind of show you the idea of how a brain-computer interface could be used to decipher these neuronal codes, these spikes of information. So the idea would be that electrodes would be implanted within a brain, and it would pick up a particular pattern of spikes across a large ensemble of neurons. So here you can see each of these little tick marks represents a spike. So you could imagine that this is kind of like a Morse code. The brain is transmitting the signals that it's representing at that time. And then that could be sent to a computer chip or a computer algorithm that would decipher these codes that contain the stored memory. And that in this way, perhaps this would allow memory to be recovered if there was a deficit in the retrieval mechanism, for example. So here you can see a more specific example of how this would take place. Let's say that we had a grandmother having a wonderful experience with her granddaughter, and perhaps this would be the pattern of spikes in the brain that would reflect the memory of her granddaughter, that this is her encoding this experience of this time with her granddaughter. And then at another time, if she's spending time with her daughter and encoding an experience, storing a memory of that experience that she's having with her daughter, then a different pattern of spikes would be seen across a group of neurons. And then the idea would be that later, when the woman again encounters her granddaughter, that same or a similar pattern of spikes would be recapitulated, and that would allow the brain to retrieve the memory of, oh, this is, again, my granddaughter. I remember that this is my granddaughter. But as far as using this type of strategy as a potential treatment for Alzheimer's disease, there remains a few obstacles that we have to overcome before we can tell whether this is a viable treatment, because we know that Alzheimer's disease patients have trouble remembering, but we don't know whether this is because they can't store new memories or whether this is because they can store memories, but then the brain has a problem in later retrieving those memories. And part of the reason why we don't know the answers to these questions is because we haven't yet deciphered the brain activity. We haven't deciphered those spikes that the brain's memory networks are transmitting in Alzheimer's disease patients. So in my lab, what we do is we use animal models in order to decipher the patterns of activity that are being represented by our brain's key memory networks, including a brain area known as the hippocampus that I'm going to talk to you about today. So what we do is we implant electrodes into the brains of rodents, specifically rats, and then we record these spikes of activity from large ensembles of neurons, and we decipher the messages that they're sending. And we use rats because rats are really smart, and the neurons in their hippocampus are very similar to the neurons in the human hippocampus. And also the anatomical structure of the hippocampus in the rat is very similar to the structure of the hippocampus in humans. So this is a little cartoon to kind of give you the basic gist of the idea. If you think about a rat having an experience and learning something that's important to a rat, which in this case would be the path that it can take to receive a reward. So you can imagine that as the animal is learning the location of this reward, learning where they have to go to get this delicious piece of cheese, the animal is going to move through the sequence of locations that together make up this path to the reward. And neurons in the rat's hippocampus are going to fire in a specific pattern that represents that sequence of locations, and together forms the memory for that path, that learned path towards this remembered location where the rat will find cheese. And so the method that we use to decode the neuronal activity in the hippocampus, the method that we use to decipher these spike patterns, is a method that's known as Bayesian decoding. And I'm going to get a little bit technical with you here, but if you don't understand this part, it's totally okay. You're still going to be able to understand the rest of the talk. But for those of you who are interested, I'm just going to discuss this briefly. It's a statistical method that allows us to determine across time what neurons in the hippocampus are most likely to be representing. So we use a technique called Bayesian inference, and it incorporates Bayes' rule, which is the following. It says, what is the probability that the animal is in a given location in the environment, X, given the pattern of spiking activity at a particular moment in time, which is N, and you solve that probability using Bayes' rule, which plugs in the probability of spikes occurring at each location. So as the animal earlier traversed that path, you experimentally measure how the cells fire at all those different locations. What is their pattern of spikes that they're firing at all those different locations? And you plug that in. And you also say, what is the probability the animal would spend time in each of these locations? That's the probability of X here. And then you have a normalizing constant, which is P of N. And that's just because this whole probability has to add up to 1. The animal, presumably the hippocampus is representing something. The animal has to be somewhere. And when you use this technique, you get these probability distributions. And throughout the rest of the talk, I'm going to be showing you plots like this, where this is a color-coded probability distribution. This is the probability here on the left-hand side is what's being represented here. And the maximum probability, or the warm colors, the red colors, I'm going to be calling that the decoded location, because that's basically telling you the most likely information that the pattern of spikes is representing at a given time. Okay, so this is some data from our lab, just showing you an example from a normal healthy rat. And one thing that we know from our research is that as animals learn something new, such as a path towards a location where they're going to receive a food reward, we see the representation of the hippocampus changing in a specific way. So before learning, in an early trial, before the animal learns where this reward location is, you can see on the x-axis here, I'm showing you time, and then the y-axis is location. So the animal is moving in this direction, and the white solid line is showing you the animal's actual behavior. So the animal is moving along this path like this, and then this red dotted line shows you the reward location, but the animal hasn't learned that yet. They don't know yet that they're going to get something yummy when they go to this place and stop there. And so you can see the decoded location, these warm colors, are pretty closely following what the animal is actually doing. But then if you look on a later trial, after the animal has learned that at this location, they are going to receive a yummy reward, what you can see is that the decoded representation, the information that these patterns of spikes are representing, is actually sweeping ahead of the animal, where the animal actually is, and predicting where they're heading towards that reward location, almost like the hippocampus is thinking about this plan to go to this location that it remembers earlier they got a reward there. And so this is another example, because something else that we've done in our lab recently that we're excited about is we've compared times when animals learn a path to a reward location and then they later remember that correctly, to times when their memory fails. And so here what I'm showing you is an example from a correct trial. And so again, we've got time on the x-axis, position in space on the y-axis, and here this white solid line is the animal's actual trajectory. This is the animal moving along, making its way towards this location where they learned earlier that this is a place where they're going to get a reward if they go there, and they stop there. And this is a correct trial, so this gray dotted line indicates the correct reward location, and it's a correct trial, so it also indicates the location where the animal actually stopped. And what you can see is that these color-coded probability distributions, these warm colors, again, are sweeping ahead towards that reward location where the animal's heading, and then it jumps back and keeps track of what's actually going on, and then it sweeps ahead again, predicting what the animal is, about to do, and then it jumps back to what's actually going on, and so on and so forth. And now we can compare this to a time when the animal doesn't remember the reward location correctly. And you can see that those kind of predictive representations that are almost like a neuronal reflection of the animal retrieving their memory for that upcoming path to take to get that reward, those representations are missing here on the error trial. So in conclusion, we think that these neuroprosthetic technologies and these neuronal decoding of memory methods offer an exciting hope for a new potential therapy for Alzheimer's disease. However, much more research is needed to test the viability of this as a therapeutic strategy, because we don't actually know what the memory networks like the hippocampus, how neurons are representing ongoing experiences and representing memories in Alzheimer's diseased brains. So we need to do more research, and this is what my lab is planning to do in the future. We need to do more research to discover how memories are stored and how they are later retrieved in Alzheimer's diseased brains. And I would just like to end by acknowledging my lab members, and the data that I showed you in the last few slides is from Qianglong Zhang, Ernie Huan, and Carlos Losa. I'd also like to acknowledge all of our other awesome lab members and our funding sources, including the NIH, National Institute of Mental Health, National Science Foundation, and the Army. And thank you all for your attention.
Well, thank you, Laura. That was really fascinating work, and it's remarkable how what we, most of us think of memory as a very qualitative thing, and you are actually bringing quantitative techniques, scientific techniques for estimating the probabilities. So let's address some of the questions from the audience, and in that spirit, one of the participants is asking, can you tell us more about the computational techniques used to analyze the data and how it could relate to Alzheimer's disease and how you would make that connection to the disease?
Yeah, so that's a really good question, because I actually, to make those schematic cartoons, I did simplify it and I showed it as if it would be okay if you only recorded from like 5 or 10 cells. But in reality, you need tens or even hundreds of cells to be able to decode what the networks are representing. And so that is an open question in Alzheimer's disease, because we know that synapses are being lost in Alzheimer's disease. We know that there's actual atrophy of these brain networks, this area that we're recording from, the hippocampus. The cells are actually gone in the later stages of Alzheimer's disease. So that's kind of just emphasizing what I was saying at the end, is that in order to get these high probabilities where you can say with high confidence that this is what the brain is actually representing across time, you need to have the capability to be able to record very well from large populations of neurons. And there needs to be more basic research done first, just in animal models of these pathologies, to see whether it's even viable. But then there's a lot of problems of employing it in the human brain. But fortunately, there's companies like Neuralink and Kernel that are investing a lot of money in some of those type of problems, like making the electrodes, less damaging to the brain, and the wireless technologies and wireless transmission of power and the miniaturization of these devices, and all of these things to make it a viable technology in humans. But in our lab, we're just really, really excited that companies are, are investing in this technology, because I think that, it just makes it more likely that the technological problems can be solved if we are having a multi-pronged approach to it.
Fascinating. Gives a lot of hope for translating this. There was a pre-submitted question that is related, and I thought it was kind of an interesting point. You brought it up earlier that there's the possibility that a memory pattern is in the brain but is not able to be implemented in the performance. And so my question is would that correlate in your experiments? Would that be something where the encoding looks great? It looks like they're doing really well, but they're not actually performing? Or how would you distinguish in your experiments between a failure to encode versus they're encoding, but they're not actually able to translate it?
Yeah, so I think that that's what's really exciting about it, because we can see the development of the memory across trials. We can see it developing as the animal is learning something and their behavior is reflecting that they've learned that because they're performing the task correctly. So in rat models of Alzheimer's disease, it may be that we won't see the memory develop in the first place. And then, to be honest, I would say that would be bad news for this type of technology. But it could also be, like you mentioned, that the memory develops and it's getting stored, and we can see it in the hippocampal activity, but it's somehow there's something that's going wrong in the retrieval machinery. And there are Alzheimer's disease researchers who believe that, at least, in the earlier stages of the disease, that might be kind of more of what the problem is. And so then that would be more promising. Or it could be that the memory is degraded such that the brain itself can't put the pattern together, because it's almost like if you have the jigsaw puzzle and you don't have all the pieces, and you don't have enough pieces there for the brain to recognize the pattern, but maybe a strong computer algorithm could be developed to do the pattern, to recognize the degraded pattern, and then so there's a couple different possibilities, but we'll really have to see what's going on in the Alzheimer's disease brain to see which things are viable and what's needed.
Fascinating. And for the first time, modern neuroscience is addressing questions that only philosophers could really address historically. So this is really great. We have a question from Dale. Have you considered the security and privacy issues associated with recording outside of the brain that may be used by third parties in the future? And I guess this relates to humans and the incredible successes that might occur with recording and decoding, but then how would that lead to the cloud, getting our information onto the cloud of things that we've thought of, but they're not really meant to be public?
Yeah, I think that it is, it's a question that we think about a lot in terms of our decoding research, because I think that there are real ethical considerations for it, in terms of, I think that, some people would say, like, oh, this is like a technology that could be used to help us remember everything, because we forget a lot of what we experience. And that wouldn't really be a direction that we would imagine. We would imagine it more as something for like a devastating memory disorder like Alzheimer's disease. But I think it is a real concern. Some people may be afraid of this being kind of like a mind reading thing. If your thoughts are being transmitted into computer code, yeah, there would have to be a way to make sure that it's protected, and it's not like people can be like, your thoughts can be transmitted, somehow. But I mean, since we're working in rats, we don't worry about it. But in terms of translating the technology, it's definitely something to think about. And that's why I personally wouldn't imagine it being something that would be used in normal, healthy people, that it would be if it would be an application for a devastating disorder where it's worth the risks that are involved.
So this is wonderful. Maybe just in the remaining 1 to 2 minutes in this session, this gets to the natural thing that's on all of our minds: a good night's sleep and consolidating our memories. Many of us do shift work. Many of the students in the audience are used to cramming for exams and staying up all night. Can you say a few words about how sleep consolidates memory and what your work reveals about that?
Yeah, so that's also a really good question. And it's also an area where I simplified. So what we know is happening during sleep, and that's work, our lab is working on that, but it's based on a foundation. I mean, all of this I'm talking about today is based on a strong foundation from many, many other excellent labs as well. But it's very well established that, like, these spike patterns that I've showed you that represent memories, that when you sleep, those get consolidated into a compressed form that doesn't have the same level of detail as the original experience, but it's the same general pattern, but it's more like the basic gist, right? Because when you remember, you retrieve memories of your experiences later, you don't act it out again all in real time. It's like you have this kind of compressed version of it that you remember what happened, and you remember some details of it, but not everything. And so that process of making that compressed representation, which is what is stored long term and which is what is later retrieved, slow wave sleep is extremely important for that process. And so having a good night's sleep, and also the quality of your sleep, is really, really important for remembering things long term. So yes, students, do not stay up all night cramming. That's not good for memory.
Great. Well, thank you, Laura, so much for this informative and interesting, exciting area of neuroscience and very practical. So I would like now to introduce John Pierce. John is an associate professor of neuroscience, and the M. June and J. Virgil Wagner professorship holds the professorship in molecular biology. John studies the genetic mechanisms that govern behaviors and contribute to neurological disease. Believe it or not, using a tiny 1 millimeter worm, which he will discuss, this simple but powerful animal model has revealed deep insights into neurodegeneration, underlying diseases like Alzheimer's. John?
Hi, so welcome. Thank you for coming. My name is John Pierce. I'm an associate professor in the Department of Neuroscience, and I'm also the father of these 3 Longhorn fans shown here. Today, I'll share with you our research related to Alzheimer's disease. I became interested in Alzheimer's when my first son was born with Down syndrome 19 years ago. You might not know this, but virtually everyone with Down syndrome develops Alzheimer's disease. So what would you do if your newborn son was fated to develop Alzheimer's? Alzheimer's is caused by accumulation of toxic peptides derived from the APP protein. People with Down syndrome get Alzheimer's because they have an extra copy of this APP gene that gives rise to more toxic peptides. APP peptides contribute to plaques in the brains, they cause dysfunction of neural circuits, and trigger tangles of toxic tau proteins within neurons. At first, neurons important for memory die, and over the next decade, a wave of neurodegeneration spreads. This impacts mood, sleep, and eventually personality and motor function. Researchers surprisingly still don't know how neurons die in Alzheimer's disease. Most people here probably know someone who has Alzheimer's disease. This is because it's the most common form of neurodegeneration and the 6th leading cause of death in the US. Sadly, there's no way to stop or cure Alzheimer's disease right now. In the US, Alzheimer's affects about 5 million people, and over the next few decades, with the growth of the elderly across the world, there are more people going to have Alzheimer's than there are people living in Europe now. Researchers typically study Alzheimer's disease with mouse models because they have similar brain structures and develop plaques just like humans. Unfortunately, the neurons don't die in the mouse models. They require a couple years to get old enough to develop symptoms of Alzheimer's. And because of some of these problems, I decided to switch to a model that has many advantages with my family's clock ticking fast. We engineered an Alzheimer's worm that's, like David just said, 1 millimeter long that carries the human APP gene, and it shows signs of neurodegeneration. Advantages for this worm model include that it reaches old age in just 1 week. You can come up with an idea on the weekend and test it throughout the week and see the answer by the end, and test it again all over again the next week. And we can screen through many drugs and genetic manipulations to test if they prevent neurodegeneration. Because worms are transparent, we simply peered through their body to check which neurons are dying. Here's a picture of a bunch of worms kind of packed in like sardines together in a row to take pictures. To make it even easier, we labeled some of their neurons green, or that you can see with fluorescent light. When we zoom in, we can see that there's a subset of their neurons that die when they reach old age of about a couple days. With the convenience of our Alzheimer's worm, we tested hundreds of drugs that might prevent neurodegeneration. Some drugs approved for human use work better than others. Interestingly, ones that provide moderate detection later failed clinical trials quicker than others. Interestingly, one novel drug that we work with here at UT made neurodegeneration worse, but that led to new drugs that we knew would hit the same target and had very potent neuroprotective effects. Impatient that we weren't working fast enough, we collaborated with Dr. Adela Ben-Yakir in the Department of Engineering here. She built a system to grow our Alzheimer's worms in multi-well plates to test many different drugs. Her system automatically sucks up the worms and scans their glowing nervous system to judge whether or not a neuron is dying or not, and the drugs are neuroprotective. So with this strategy, we can aim to test millions of different potential drugs. 5 years ago, I received a shocking surprise. I teach a neurogenetics class, and I have the students studying my genome with 23andMe. As a class, we learned that I carry an unlucky version of a gene that predisposes me to Alzheimer's disease. The APOE4 gene gives me a 90% chance of developing Alzheimer's in maybe about 15 years. Coincidentally, just as I learned this news, my mother turns mid-60s and developed Alzheimer's. So our research became much more important, more personal, moving from my cause trying to care for my son to worry about myself and my mom and all of my future progeny, I suppose. This is, scientists don't know how the APOE4 gene leads to Alzheimer's disease and makes it happen faster. This is despite the fact that the vast majority of people with Alzheimer's carry versions of this gene. To study how APOE4 worsens neurodegeneration in Alzheimer's, we added the APOE4 gene to our Alzheimer's worm. We were impressed to see that the APOE4 gene by itself had hardly any effect, but when we combined with the APP gene from humans, it caused more severe neurodegeneration. In fact, you can see that here with the neurons that are color-coded green compared to control. When you combine the two together, the neurons completely disappear after they die. Without this neuron, baby worms end up hatching inside because this neuron has the role in opening up a hole to lay eggs. So neurodegeneration is apparent by this alien effect of the baby worms hatching and exploding out of the mom. Although grotesque, this makes it really easy for us and also undergraduates to help us identify chemical compounds and genetic manipulations that can lead to neuroprotection. We still don't know how neurons die in Alzheimer's disease. To solve murders, police interview eyewitnesses and perform reenactments. Luckily, with our worm model, we can reenact and witness neurodegeneration every day. Here's a movie of a neuron dying made by my postdoc, Lottie Brose. So initially, it's healthy and green. As time goes on, APP that's tagged red accumulates in certain portions of the cell. And you can see that that leads to the neuron stretching off and trying to release parts of this. Over time, it dies. This form of death is called necrosis. And consistent with this theory, we found that deleting genes required for necrosis prevents APP and APOE4 from killing the neuron. These are some of the first evidence that shows how neurons die in Alzheimer's disease. Another major mystery in Alzheimer's is why some neurons die while others stay alive. And why does it start in certain areas of the brain, despite the fact that these disease-causing genes are found throughout? Perhaps some neurons have vulnerability factors, while others have factors that help them survive. To test this theory, it'd be great to know what factors, what genes are found in every neuron throughout the body. Fortunately for us worm researchers, we have a list of this. So for every neuron throughout the body, there's a chart of what gene they express, at what level. And if we compare the level of genes in the surviving neurons with those that degenerate, we can get candidate factors for vulnerability and protection. Now we're in the process of empirically testing whether or not these genes are required or sufficient for protecting or vulnerability of these neurons. I want to extend thanks to many undergraduate volunteers who've helped with their research, as well as my lab staff. We're very appreciative of the funds that we've got from NIH, private foundations, and individuals from alumni like you guys who are watching in the audience. I have a special Shout out to George and Karen Casey, the Pine Foundation, and David and Ellen Bertman. Thank you very much for listening. I'm hoping that we can make a big difference in leading to drug discovery to treat Alzheimer's disease.
Thank you, John. That was for that fascinating review and overview. I mean, what goes through my mind is the how fundamental discoveries can be made in simple organisms much more quickly. And your personal story is really very compelling and appreciated. We have some questions from the audience that we'd love to present to you and for discussion. Teresa asks, if you can test for the ApoE4 gene, can you destroy it if you test positive for it, or can you inhibit it? So maybe we'll go with that question for now.
Okay, that's a good question. Right now, we're in this new era of personal genetics, where you can go ahead and use 23andMe or some other company to kind of take a peek at your genome and see definitively whether or not you carry an unlucky version of certain genes. Every one of us carries the ApoE gene, and if you have 2 tweaks on the gene, that defines the 4th version that predisposes you to high risk for Alzheimer's. You wouldn't really want to get rid of the gene itself. It's doing important things like transporting cholesterol and lipids throughout your body. But there are scientists out there who are looking for drugs that can mimic kind of the healthier versions of ApoE. There's one called ApoE2 that leads to lower risk of Alzheimer's. The Vivek lab is making drugs that can try to transform the ApoE4 version and bend it into like an ApoE2 version, a healthier version. I hope that answers your question.
Yeah, George asks a very interesting question in this modern discussion of vaccines for COVID with mRNA and DNA-based vaccines. Could a vaccine be developed against the gene or the set of genes that we are interested in targeting? How would one think about that?
That's another great question. I think all of us it's breathtaking what this mRNA vaccines can do with their efficacy and also their speed of development. That gives scientists outside of the field of virology kind of excitement that we might be able to target dangerous proteins that are in our own body that contribute to disease. For Alzheimer's, people have already been developing approaches to reduce the amount of toxic peptides from APP, including A-beta, that contributes to plaques in the brains of people with Alzheimer's. That would be an interesting place to start. However, we have to be cautious because scientists have succeeded in coming up with several drugs that can erase plaques in the brains of people with Alzheimer's. And sadly, the people still go on to die and their memory remains declining. So ApoE4, other versions of APP, there's another toxic protein called tau. There's a number of good candidates that researchers are investigating to see if we can target them, immobilize them, and render them inert.
We have a great question from Falcon, which raises a broader question, which is maybe if you could broaden the discussion about use of simple organisms to solve degenerative diseases. What are some of the other examples that have been where success has been made using simple organisms, and why are simple organisms advantageous other than the fact that they age quickly, as you gave in this example? And is the worm that you used a different animal, or did you have to genetically modify it? So a little discussion on the model itself.
Yeah, we're not the first people to study how neurons in cells die in an organism using this tiny worm, C. elegans. In fact, the 2003 Nobel Prize was given to a collection of researchers, two of which discovered how cells die naturally during development. When we're all embryos, we have extra cells that need to be eliminated during the course of development. If you have problems with this pathway, you'll end up being born with webbed fingers and toes and have some other problems. In a severe case, if not enough neurons die during development or birth, you can have, the brain gets so big that you can have problems when the babies are born. Now with C. elegans, you can stop cell death and they can have an extra 100 or so cells. They don't have a skull and bone structure constraining them, so they can live happily. So they can also live with some severe mutations, even that render them immobile or unable to find a mate, but they're hermaphroditic, so they can continue to reproduce. So we can still study severe mutations that would be lethal in any other type of animal. So major discoveries in other cell death pathways like necrosis, some new ones that you may not have heard of, have been made with this tiny animal, thanks to these properties, especially its transparent body, where you can see every one of the cells, which are all accounted for with by unique names.
Thank you. Another question that's come up is, is there any evidence to suggest that viruses play a role in Alzheimer's? And what are your thoughts on a viral basis for it?
That's a great question too. One of the hypotheses behind the origin for Alzheimer's disease might be that as we get older, there might be chance infections that get past the blood-brain barrier into the brain. They could be viral, they could be fungal or bacterial, and the brain will try to protect itself. In fact, there's decent evidence that shows that the plaques themselves might be protective. They might, the brain might release these and try to surround a pathogen to immobilize it and render it inert. So, but if this happens enough, or if this happens in the wrong people with the certain genetic backgrounds, that might be what triggers Alzheimer's disease. There's a number of other genes that are in our immune pathway that, if tweaked, can modestly increase risk for Alzheimer's disease. So those two types of experimental evidence do, support some of the viral or fungal or the pathogenic origin for Alzheimer's disease.
Well, thank you very much, John. I'm sure there are other questions coming from the audience. We have a very curious audience, which is great, and please feel free to submit more questions through the communications office. I wanted to thank you, John, and for joining, and all of you for joining us today for our Texas Science Festival talk on addressing Alzheimer's. And I'd like to thank John and Laura for their time, knowledge, and expertise, and for all the great questions that have been asked from the audience and addressed by the speakers. Thank you again. We do have a message here that follow-up questions can be sent to cnsdev@austin.utexas.edu. So feel free to continue the discussion by email. Thank you very much.